Hello students, welcome to another lecture Understanding Biology chapter 31. In this lecture we will talk about plant growth responses to different stimuli, environmental stimuli. At the end of this lecture you should be able to compare the pigment called phytochrome and chlorophyll, describe growth responses influenced by phytochromes, describe the mechanism of phototropism, and describe how plants may perceive and respond to gravity. So let's look into how plants are responding to light first. Previously, you learned about photosynthesis, in which plants convert light energy into chemical bond energy that they store. This is possible because of the photosynthetic pigment called chlorophyll. Apart from chlorophyll, which is actually involves in photosynthesis in plants, there are many other pigments that involve in detecting light and mediate plant responses to light. So, light actually can influence the seed germination, flowering, and other critical developmental events in plants. So, it is very important for a plant to detect the light and respond to it accordingly. So when a plant exhibits non-directional light triggered development, it is called photomorphogenesis. When a plant exhibits a directional growth response to a light, it is called phototropism. So these are two different types of growth responses to light. One is non-directional, the other one is directional growth responses. So non-directional one is called photomorphogenesis and the directional one is called phototropism. So both of these type of responses actually indicates that plants don't have the ability to move away from some sort of not good environmental conditions. So they have to either respond to it or adapt to it. So this indicates both of these situations, photomorphogenesis and phototropism, actually indicate that plants are unable to move away from these unfavorable conditions. So let's look at how this extra pigment that we are going to talk about, phytochrome first, is actually involved in responding to light. So phytochrome is actually an important pigment that involved in plant growth responses. This pigment is present in all groups of plants and in a few groups of green algae, but this pigment cannot be found in protista, bacteria, and fungi. So it's actually found in all groups of plants. So phytochrome molecule, when you look at the molecular structure, it actually shows two interchangeable forms. One is PR, which actually absorbs the red light at 660 nanometer wavelength, and we have PFR, which absorbs the far red light at 730 nanometer. Apart from the phytochrome protein, when phytochrome gene is expressed, the protein that is produced actually have two parts. A smaller part which is called chromophore. Chromophores are actually sensing the light, and then we also have a bigger portion that is facilitating the expression of light response genes called apoprotein. So we have a smaller portion and the larger portion. The smaller portion is actually sensing the light, and larger portion, which is called apoprotein, actually help to express the gene responses. Phytochromes are actually involved in numerous signaling pathways that are responsible for number of plant growth responses. These include seed germination, shoot elongation, and detection of plant spacing. So we are going to discuss one by one how phytochrome is actually involved in each of these growth responses. So let's first look into seed germination. Seed germination is actually inhibited by far red light. Far red light, if you remember, it is at 730 nanometer, and the seed germination actually stimulated by red light, which is 660 nanometers. In many plants this happens. So you know that chlorophyll actually absorb the red light, and when you think about a plant, the canopy is actually where the leaves are, so chlorophyll will actually absorb a lot of red light from canopy, and so above the seed, these canopy actually contain and trap all the red light. So by the time that it get to the seed level, it largely reduce the amount of red light, and only the far red light is available. Here is an example of why it is that the seeds are actually needed the red light for their germination. Sometimes seeds of these deciduous plants, seeds that are under these deciduous plants, which mean the deciduous mean that these plants will lose their leaves during the winter, and then they will reproduce and reproduce their leaves and come back in springtime. So when there are seeds under the soil, under these deciduous plants, they will not germinate throughout the summer, because as you know, the leaves will absorb all the red light. So the seeds actually need direct red light to germinate, so they will not get a chance to germinate until the winter has passed and they lost all of their leaves, and in the spring they will get direct red light, and this is the chance for them to germinate. So that is about the seed germination and how light is actually influencing the seed germination. And then the next one we are going to talk about is shoot elongation. It is another response controlled by phytochromes. Elongation of a shoot in a seedling that kept in a dark is caused by lack of this red light. So this is called etiolation. When you leave a seedling in a dark room, it is actually prematurely get very long. So this is an adaptation of that seedling without light to make it taller until it reach light at some time of their life cycle. So this phenomenon is called etiolation. So the hormone, actually there is a hormone that play a role in plant responses to light through phytochromes. This hormone is called brachiosteroids, and the shoot elongation also require direct red light. When the red light is lacking, they will just not properly elongate. So lastly, red and far red light also involved in signaling the plant spacing. So plant spacing is also very important because when plants are too close to each other, they will compete for light. So if a plant can sense how far away it's or how closely the neighbor is located, they can actually increase their height to reach maximum light. So usually when plant is too close together, leaf shading can happen. This leaf shading increases the amount of far red light compared to the red light. Plants can measure the amount of far red light reflected from the neighboring plant, and if the neighbor is too close, plant will grow taller. So that is how these responses are controlled by light and phytocyanin. Now the next one we are going to talk about is phototropism. As you remember, we defined phototropism as the directional growth responses to light. So phototropic responses include the bending of growing stems towards the sun. In generally, stems are positively phototropic. Phototropism is usually adaptive feature of plants because they want to get more light. So this adaptive value is very good for the plants to receive more light, available light, for them to do photosynthesis. So another environmental stimuli plants response is called gravity. When a potted plant is tipped over and left there for a while, you might have noticed that the shoot actually bends and grows upward. This is an example of gravitopism. It's a response to the gravitational field of Earth. So gravitopic responses are present at germination when root grows down and shoot grows up. Therefore, shoot have negative gravitopic response and root have positive gravitopic response. So there are 4 general steps that lead to gravitopic response. So these are actually, first the gravity is received by the cell and a mechanical signal is transduced into a physiological signal, and physiological signal is transduced inside the cell and to other cells, and differential cell elongation occurs in the up and down side of the root and shoot. So this is how actually plants are responding to the gravity. So currently researchers are debating the steps involved in perception of gravity, because in shoots gravity is sensed along the length of the stems and endodermis cells, and signaling occurs towards the outer epidermal cells. But in roots the cap is the site of gravity perception, and a signal must trigger differential cell elongation zone and division zone for the root to elongate towards the gravity. So there is, as you can see, so the signal is received by the root cap cells, and the morphological differentiation, cell differentiation, is actually happening in the zone of cell elongation zone in the root. So how do these signal from the root cap cells to the elongation zone is sent is a questionable remark that scientists are currently trying to answer. So in short term, in both shoots and roots, there are these plastids called amyloplasts. So these plastids contain starch, and they actually sink towards the center of the gravitational field and may be involved in sensing gravity for both the shoots and the roots. So in roots, gravity sensing cells are located in the root cap, and like I mentioned before, the cells that actually undergo the asymmetrical growth are located in the elongation zone, which is closest to the root cap, by the way. So like I mentioned, the scientists are trying to figure out how the signal is transferred from the root cap to the cell elongation about the gravity, and they think that their auxin might have some effect of sensing the gravity and sending the signal, but they have conducted experiments without auxin and with auxin, and they still found out that even if the auxin transport is suppressed, this gravitational signaling still happens. So it is unclear how roots are responding to gravity, exact mechanism, signal transduction for root response for the gravity. So that is my last slides. In summary, plants grow and develop in response to environmental signals. Phytochrome, a red light receptor, acts through the pathways that control the gene expression. Phytochrome influences seed germination, shoot elongation, and plant spacing. Gravitropism is another environmental stimuli where plants are responding to gravity. In endodermal cells of shoots and root cap cells of roots, aminoplasts settle to the bottom, allowing the plants to sense the direction of gravitational pull. I hope you enjoyed this lecture. Until we meet from another, have a good day. Thank you.